Hello, my name is Alina Renz. I'm from the University of Tübingen in Germany, and the title of my poster is FBA reveals guanylate kinase as a potential target for antiviral therapies against SARS-CoV-2. We all know about the current spread and also about the threat the novel coronavirus holds worldwide. Now, first promising antiviral therapies and vaccines are under discussion, and first clinical trials are conducted. However, so far, no one knows when the first approved treatment or vaccine will be available. So, further research is of high interest and of high importance. The released viral nucleotide sequence facilitates both the investigation of potential antiviral targets and the research on the molecular mechanisms and metabolic changes upon infection. Those changes can, for example, be investigated by analyzing integrated host virus genome-scale metabolic models. In genome-scale metabolic models, metabolites are connected via reactions, and those reactions are catalyzed by enzymes, which in turn are encoded by genes. We used an extensive model of human alveolar macrophages, which was already published in 2010, and we incorporated a viral biomass objective function of SARS-CoV-2. To generate this viral biomass objective function, we adapted and extended the pipeline suggested and published by Eller and his colleagues in 2018. With the annotated nucleotide sequence of the virus, we calculated the stoichiometric coefficients for the nucleotides, the amino acids, and the energy requirements. Relevant for these calculations was furthermore the ratio of structural versus non-structural proteins. Since this number was not known for the novel coronavirus, all subsequent analyses were conducted with varying ratios. First analyses of the stoichiometric differences in amino acid and nucleotide usage in uninfected and in infected cells indicated differences in their requirements. Some amino acids like asparagine, tyrosine, threonine, isoleucine or phenylalanine are required more upon infection. In contrast, histidine or glutamic acid are less required in the infected cells. All 4 RNA nucleotides are required much more in the virus-infected host cell. These stoichiometric differences are first signs for potential changes in the host's metabolism and targets for antiviral therapies. In the next step, we used flux balance analysis, short FBA, and reaction knockouts to investigate the actual metabolic changes. Each reaction was knocked out in the uninfected and the infected cell, and the effect of the knockout on the host and the virus biomass function was compared. The aim of this approach was to identify enzymes whose complete inhibition do not harm the host cell but disturb the viral growth. This analysis revealed the guanylate kinase as a promising antiviral target. Its complete inhibition decreased the viral growth while not affecting the host cell. The guanylate kinase is part of the purine metabolism and converts ATP, so adenosine 3-phosphate, and guanosine monophosphate, GMP, to ADP and GDP, so adenosine and guanosine diphosphate. Since antiviral drug targets might not completely inhibit its target, the so-called host-derived enforcement analysis was conducted. Flux variability analysis, short FVA, was used to determine the range of allowed inhibition of the reactions that does not harm the host cell. Instead of totally blocking a reaction, like it's done in the reaction knockout, one might just decrease the activity of the enzyme to, for example, 50%. Again, the guanylate kinase was reported along with further targets from the purine metabolism. Amongst these further targets were the adenylosuccinate lyase and the IMP dehydrogenase. The adenylosuccinate lyase converts adenylosuccinate to AMP and fumarate, while the IMP dehydrogenase catalyzes the NAD-dependent oxidation of IMP, so inosine monophosphate, to XMP, xanthosine monophosphate. And this reaction is the rate-limiting step of the de novo synthesis of guanine nucleotides. So it seems that the purine metabolism might harbor potential targets for antiviral therapies. Thinking back to the beginning for the analysis of the stoichiometric differences, the relevance of the nucleotide metabolism was verified in the knockout and the host-derived enforcement analyses. Inhibitors of the guanylate kinase are already described in literature. These inhibitors need to be tested now in vitro experiments to verify their potential therapeutic effect against SARS-CoV-2. Further studies already mentioned the IMP dehydrogenase as an interaction partner of SARS-CoV-2. With the increasing knowledge about the current coronavirus, we can improve our viral biomass function and incorporate it into the already existing model. We can also curate further genome-scale metabolic models of relevant host cells with the virus biomass function and analyze the new host virus models to verify the role of the guanylate kinase as a potential antiviral target. With this approach, we were able to not only give some insight into the molecular mechanisms and changes upon infection, but also to identify potential drug targets. With these metabolic insights, we aim to contribute to fight against this pandemic. Thank you very much.